Thank you, sir. Hello, everyone. Good morning. My name is Hu Ge. My Chinese first name is Hu, which sounds like Hu in English. So you may call me Harry instead. And I'm very thrilled to be here to present and share how big compute is optimizing biologicals design workflow with auto-tuned models and multi-agent systems. Biologics therapeutics improved the curement of numerous diseases, but the design of biologics remains a complex task. Therefore, we aim to revolutionize the design workflow of biologics. We have built this AI-empowered design workflow, covering almost everything from antibody humanization to de novo generation and sequence redesign through structure prediction, affinity maturation, all the way to druggability, prediction and optimization, and down to wet lab validation and auto fine-tuned models. In this workflow, we have integrated many proprietary models and some of the powerful open source models and frameworks from BioNeMo. Before we move to the technical architectures of the workflow, let me show you how we can achieve through this workflow. This is a recent case study of a multi-objective design of a bispecific antibody. In this project, we designed about 50 variants and tested them in 2 rounds of design validation cycle. And we have successfully optimized 3 key properties for these variants. And we have the results are striking. We have improved the expression yield for over 11 times. And we pushed the SEC purity of the molecules from 60% to 96%. And the affinity simultaneously improved for 10 times. So how could we achieve that? We achieved that through optimizing the workflow of therapeutic biologics. For today's session, I will divide the whole workflow into 3 core tasks, and I will guide you through each of them. The first one is molecular generation, and the second is immunogenicity prediction and optimization, and the last is model fine-tuning. Now let's start from task 1, de novo generation of antibody, which is a new paradigm of antibody discovery. In the traditional antibody discovery method is like a marathon. It takes 6 to more than 12 months to complete. And the bottleneck is you need to do animal immunization, and you have limited target scope. But for the AI-driven de novo generation paradigm, it only takes about weeks, and you can directly optimize for better drug abilities. And also, you can do epitope-specific targeting. So behind this, we have developed an engine which is named Biologics. It's a high-throughput generative AI for the de novo antibody design. This model is it learns the joint sequence and structure embedding, and it is conditioned on the target epitope, which means it is target awareness. And also, we have a powerful latent geometry engine inside it, which means we model the backbone of the antibody explicitly, but we compress the details of the side chains into the latent space, so it can be sampled very efficiently. And we use an ODE-based sampler to do efficient mathematical sampling, so that we can deliver many antibodies massively. And this model can be run on a single RTX GPU, and it can deliver molecules, 3,000 molecules in 1 hour. This is an animation of the generation process of Biologics. And we compared Biologics with several open source baseline models. They are all well-known models. For example, RF antibody, IgGM, and BOSGEN. And we tested those models on the same system. And in the test of generating VHH nanobodies de novo, we can see that the speed up of Biologics over existing baseline models is about 3 times to from 3 times to about 33 times. But only generating a binder is not enough, because biologics may encounter immunogenicity problems. When the biotherapeutic is administered, firstly, it may be uptake by dendritic cells and presented to T cells. And the T cells may be activated and release cytokines to activate B cells. And the B cells may produce anti-drug antibodies. And those anti-drug antibodies, which is ADAs, they could cause problems. That is, the body will identify the biologics as intruders and eliminate the drugs. And the consequence is that the ADAs bind to your, for example, antibodies and block it from reaching the target, and rapidly clear your therapeutics from the system, and reducing the drug's half-life. And also, even worse, it may cause hypersensitivity or other adverse outcomes. Even humanized or fully human proteins can trigger these reactions. That's why the prediction is critical for FDA or EMA alliance compliance. But the legacy tools, for example, NetMRC or others, they are they only model the interaction between the MRC and your peptides. They miss the gap. They miss the biological complexity of the immunogenicity. The immunogenicity is primarily driven by the T cell dependent pathway from the uptake of the drug by the APC which is internalization, to presentation by the MHC molecules, and the recognition by the TCRs, and the B cell activation and the release of ADAs. It's very complex. So we also need a complex system to mimic the immune response. That's why we built VADAPT, which is a revolutionary system for the in silico prediction of immunogenicity. And it is a multimodal system mimicking the T-cell dependent pathways to predict ADAs in the therapeutic biologics. It is consisted of several modules. It predicts the binding of the peptide fragment of your biologics to the MHC molecules. And it also predicts the probability of the peptide of your antibody being processed and displayed on the APC cells. It also simulates a global population using representative HRA allele ensembles. And it also mimics the immune tolerance mechanism, which means it predicts if your therapeutic biologics will be treated as self or non-self intruders. So we compared our system with existing tools like NetMC, IDB, and Epimetrics, and we found that it showed a leading prediction accuracy and a superior correlation with clinical ADA incidence rates. For example, we have higher area and ROC and accuracy and FE values in the enrichment test. And we also have better correlation with clinical ADA incidence. So if our biologics are predicted to have immunogenicity risk, can we do something to fix that? The answer is yes. To reduce the risk of immunogenicity, a simple strategy is to do or introduce mutations on the T-cell epitopes that may cause an alarm. But the problem is that the mutations may cause structural instability or even render the loss of affinity of your molecules. So a solution is that an ideal optimizing system can reduce T-cell epitopes or immunogenicity scores and at the same time preserve the protein quality or folding stability or even affinity, which is a dual optimization task. So we have developed the immune MPN, which is model system that it can solve the immunogenicity and the function trade-off. It works by 3 parts, and the predictor, which we mentioned, is VADAPT. It can predict the T-cell epitope and the immunogenicity scores. And the protein MPN model, as a generator, it can propose viable mutations based on the reference structure of your antibody. And also we have a navigator, which is Monte Carlo tree search. It can manage the search space, and it can balance the exploration and exploitation. This immune MPN can intelligently navigating the massive possible mutational space very efficiently. The potential mutational space is very vast. If you use d-immune MPN, you do not have to brute forcing all the 20 to the n number of sequences. You just need to leverage this system to find the best path to your solution. And based on GPU acceleration, we have over 100 times speed up, which means you can finish the task of de-immunization for one molecule in hours. So what if we have some other domain tasks not limited to immunogenicity or other common tasks? Pre-trained models are rarely perfect out of the box. And if we have some, for example, a few in-house data, we can use few short learning to bring some massive domain improvements. And the traditional fine-tuning process is cumbersome. It requires the trial of different fine-tuning strategies and hyperparameters. So we have built this end-to-end model optimization pipeline. It leverages pre-trained protein language models such as ESM2, and it does automatic fine-tune via BioNeMo frameworks, and it can try different strategies. For example, standard 4-parameter or LoRa fine-tune, or other different task-specific heads. And it will split the data set automatically and test them one by one, and rank the models and deliver the best performance model. And built with the BioNeMo framework, our system explores all those different strategies, adjusts all the parameters, and all can be done with minimal human intervention. The user just need to upload their data in a CSV file, and it does training and fine-tuning automatically, and output a model file. And using that model file, you can do predictions on new samples immediately. BioNeMo enables access to a wider array of NVIDIA-optimized models and fine-tuning capabilities. Especially in this work, we leverage the foundational models and data loaders and optimizing and training capabilities of BioNeMo. So here I would like to thank NVIDIA and the BioNeMo team for contributing such great tools to the open source community. And here is a case study of antibody thermal stability prediction. We use 700 antibody sequences and DM values in a CSV as input, and we tested different, for example, pre-trained ESM2 models and the auto fine-tuned models. The result turns out that the fine-tuning boosted the prediction accuracy from 0.269 to 0.692, which is very significant. All right, now that I have shown what WeMo can do, I will hand it over to my colleague, Dr. Li, and he will show show you how agentic AI will take the platform to another level.
Thank you. All the nice work that Harry shown in this presentation is based on our platform, which is called Wemo. And it has a lot of features and very easy to use and has been adopted by many users, including biotech and also research lab. Now, as the AI advancement is so fast, we see the new opportunities. And so how we can make the Wemo platform more intelligent, more flexible, and more robust? And so we can take a agentic approach for this. And the first step, how we can do that? So the first step is to make all those tools into AI ready. So what do you mean, make AI ready? So as you can see that our tools, it has full spectrum from small molecules to biologics, and from the protein to antibody, and even include traditional Chinese medicine. And so what is the best strategy to make all those tools work coherently, and in the meantime to leverage the modern AI? So as I mentioned, I want to make AI ready. What does it mean is that for every tools, we want to wrap up into a certain form, let's say MCP servers. And another way for doing this is that we create a lot of agent skills, which describe all of those tools and how to use them. And in addition to that, we have vast scientific publications. It contains very rich knowledge and know-how. So we want to make the knowledge also accessible to AI tools. So that means we want to make all those tools, scientific engine, and scientific knowledge more AI accessible. That's what we mean, how to make it AI ready. So and a number of strategies we applied. So in addition to that, we also have close collaboration with NVIDIA, particularly the BioNeMo team. And we want to include a lot of tools. And in addition to collaborate with NVIDIA, we also collaborate with ChatMo team, which is an open source team. And we can integrate a lot of open models from BioNeMo into our system. For example, the Mogen. This can be used for de novo design, link design, and the need optimization, scaffold decoration, and so on. And another example is that the NV Kit is essentially a highly optimized RT kit. And even though it's targeted for GPU optimization, and even for a CPU version, it's much more efficient, up to 10x performance. Of course, for GPU, it's more dramatic, for certain calculations up to 3000x. And for the another example is for MMFF optimization, it's 24 times faster. So we want to take advantage of all those nice tools developed by NVIDIA. And in addition to those individual tools, we also have a large collection of well-designed, predefined workflows. For example, the MT simulation. Usually, this is very highly complex, procedures, include protein preparation, protonation, and set up, for example, GROMACS, simulation parameters, and also the how to do the results analysis. Data analysis is also very crucial, and we wrap up this in a very nice protocol so that it's easy to use. And so we want to also make those predefined protocols AI accessible. And so here's our design. And apparently, a very crucial issue for design is the security. And so we put this, at the very beginning in our design. So in our system, the Waymo Copilot has 4 major components. The first is the agentic execution. And the second is the sandboxed tool execution. So for each user session, it has its own sandbox to do calculation or file operation, whatever. And of course, we have a very professional security control for that. And of course, we have a LM adapter, which you can plug any LM models. And of course, the most important part is the tools and the skills, and also the knowledge base. So here is an example how we built a knowledge base. From left to right, we start from the document. Document processing, is PDF. We'll convert PDF into text, and most likely is Markdown format. And of course, the context is in structured format. And so we build a content tree. And we also do the enrichment for those documents, and like create a summary, create a sentence embedding, and so on. And of course, we build an index, dual index, both semantic and lexical. And we use a hybrid retrieval method to retrieve content. Of course, you can, based on the efficient content retrieval, you can create your answer. And so the best way to show the power is, take an example. So now, in this example, we do the novo antibody design by, driven by the agent system. So we choose a target. That's a protein one called 1-ALY, and that's in the middle, the picture in the middle. So we use the Paradex, that's the tool Harry mentioned, to generate the antibody. And so on the right side, it shows the complex structure of the target and designed antibody. And of course, one crucial important thing is that immunogenicity risk assessment. And so, if immediately after the design we do the assessment, and in this case it will show it has high risk. In our scale, 0.39 it's high risk. 0.2 is bottleneck. So in this case, it has high risk. So we want to modify it. So we are using the immune MPAN module to do the optimization. It's based on the assessment of immunogenicity risk and also Monte Carlo search. So we mutate and we do the assessment and mutate and so on. So by a lot of iteration, we can generate, optimize the antibody, make sure it has same functionality and also have the same structure. So it's a constraint optimization. And so it'll go through the optimization, and here is the results on the table. So on the third column, it shows the immunogenicity risk. So at the beginning, the original one has 0.39. And so we have let's say top 5 candidates, invariants, and it shows the afterwards what is the risk. So right now it's reduced to below 0.1. Below anything below 2.1, it means it's pretty safe. So so after this, we get pretty good candidates. So the only thing left here is we haven't done the wet experiment yet. So that will be our next step. So just to recap, I have a video here. So it has 5 steps. Potentially, it can all be done by the agent system. So first, set up the run the de novo generation, and on the left side is the target, is the antigen protein. And so we go to the next step. This shows the 3D structures of the target and the designed antibody. And because we after that we do the immunogenicity risk assessment, and we take the sequence and dump into the Wemo Copilot, and it will automatically run the assessment. So because we find it's high risk, so it automatically trigger the optimization, which is called de-immune MPNN. And this job takes about 1 hour because it's a very intensive computation, Monte Carlo search. So after 1 hour, we just ask the system AI to analyze my results, and it take the computational log and organize into a nice table. So this is the table automatically generated by the AI. So that's the full cycle, end-to-end work done by our agentic system called Wemo Copilot. And if you want more details about our company profile, here it is. We have been in the area for over 10 years, and we have more than 500 global partners, and we completed more than 1,000 projects for different clients. And of course, we have a lot of publications, and also a lot of technology here is based on our publication, and also we have a lot of publication results from our tools. And if we want to learn more, if we want to give a try, here is the website. It's wemo.net, and it's free. You can you use your email to register the account, and then try it out. Yeah, that's all. Thank you. And now we are ready to take questions. And I will invite Harry and also our another colleague, Roy, to join the Q&A. Thank you.
Any questions from the audience? Sure, come on up to the microphone. Maybe while he's coming up, can I ask how are you thinking about the evolution of your platform and agents as a service, or your open claw strategy, for instance?
Yeah, it's moving so fast. It's very hard to catch. So immediately after Jason Wang's keynotes, I communicated with my colleagues in China, and my colleague asked, what's the news? I talked to them about open cloud and Nemo Cloud, and Nemo Cloud is released immediately after the talk. So my colleague immediately opened his computer and opened the GitHub and search for the Nemo Cloud and try to analyze it. I try to learn it. So I think the key issue here is the safety. So because the Nemo cloud, put the safety in the central. So I think it's a good starting point for us, to learn more and collaborate with NVIDIA more and to move forward. And hopefully we can make a contribution to the society. Yeah.
Go ahead.
Hi, this is super interesting. So as you know, there's been a lot of work in the antibody design space, and I know people are generally interested in deimmunizing or alternatively humanizing these antibodies. And so there's been a few papers published by like Charlotte Dean from Oxford and a few other groups that have identified like key regions of antibodies that contribute to stability or reducing immunogenicity. I was curious if you noticed any correlations between your models and these existing works, or are they kind of uncovering new residues that are crucial for reducing like MHC binding?
Sorry, could you be a little bit slowly?
Oh yeah, sorry.
Especially for the last few sentences.
Yeah, sorry.
Yeah.
So there's been a lot of work done on humanization.
Yeah.
I'm wondering if your model has pulled out the same residues that existing work has shown, or if you're uncovering new residues or amino acids that are suggesting areas that improve stability or reduce immunogenicity.
Yeah, we basically introduce new mutations onto the region where it's predicted to be a potential TCR epitope. But when introducing mutations, you can do that randomly. And also you can do that, for example, using fragments from human libraries. And you can also using, for example, MPNN. In that case, your mutations will fit the structure better so that it will not influence or be harmful to the folding or even affinity of your antibody. Yeah. But if you use human fragments, then it's like humanization. If you use mutations from MPNN, it will be maybe alien residues. Yeah.
Great, thank you.
Thank you.
From our experience, I think the most dangerous is region is cross the FR region and the CDR, just the adjacent region. I think because the FR is from germline, it's usually the same, but mostly either cross the FR and the CDR agent region or within the CDR region. Yeah.
Cool, thank you.
All right. Well, thank you very much, Harry and Giabo. This was an amazing presentation. I learned so much about WeCompute and your platform, and I hope everybody goes and checks it out. So thank you. Enjoy the rest of GTC.